Okay. Assalamu alaikum. Can we start, everybody? Morning. I think today his strength is less. I think maybe yesterday's dinner was very heavy. It looks like. Okay. Okay. Because I have to leave soon, so I have to start. I have a couple of slides, maybe 10, 15 slides. I want to give you some thoughts. So, for last 4 days, we have gone through what is the drug discovery and how we connect as an academic institution with the industry. That will be few thoughts. I will give you some reading material. You can get it from them, and these are not the drug discovery reading material, but science fiction kind of thing. But it is true, and I will explain to you, inshallah. So these are the 3 books which I have read. Okay, so one is Drug Hunter. I think if you can get in your library here, that will be really nice. So this is written by दो जर्मन साइंटिस्ट ने लिखा है इसको, और कैसे जो है ड्रग डेवलप की जाती है, ढूंढी जाती है, और उसके बाद मार्केट की जाती है. दिस इज़ ऑल, एवरीथिंग इज़ देयर. This was written by from 2 German scientists from Schering AG. Second book is very famous, The $1 Billion Molecule. Okay, and this was written by this was actually written by a regular writer, Barry Rith, but this is the story of Dr. Joshua Boger, who was working at Merck, and then he went out and made a company called Vertex, which is like about $80–90 billion company. So this is the life story of Dr. Joshua Boger. And how many people know about Venki Ramakrishnan? Anybody knows about Dr. Saab? Yeah, see, he knows. So, Nobel laureate. So, yeah, and the biology problem. So, gene machine. Venki Ramakrishnan is a senior scientist in Oxford, and he still writes. He don't have a car. He still rides his bike. So he's so famous. Very nice book. And you really enjoy reading at least 2 of them. Third one, if you don't want, I think you can ask your library to get all these books. Okay. So this will give you a glimpse from academia to industry, because Venki is working in academia, and he has done a lot of work on ribosomes, which is a organelle in the cell. And $1 Billion Drug is, $1 Billion Molecule is a industry book, and the first one is both industry and academic. So try that sometime. Okay. So what we talked all along, last 4 days, right? We talked about the process of drug discovery, and then how it is going in academia, and what are the important part of discovery development. So we learned target discovery. We have given a lecture. Then hit generation, lead generation, and this is called, we call it exploratory. Just I'm capping up so that you understand what we talked. And then candidate, we select a candidate. We go to NL model. Dr. Zainuddin talked about all those things. Lead optimization, I talked in my own area. Then clinical trials, we have given several examples of clinical trials. And then development, and then it goes to the approval, and then post-approval. So this is the complete process of drug discovery. If you want to read more, there is a book there. Okay, down, Deline et al. In book Introduction to Biotech Entrepreneurship from Idea to Business. So you are going to have idea. We discussed yesterday a lot of ideas. If you want to know how to go to business, read this book. Okay. And Dr. Syed was here. He came from a lab to entrepreneur. He has about 300 crore business now. So that I'm sure that he read this book also. Okay. Okay, so there was I have 5, 6 more slides, and this was I wanted to tell yesterday, but we went into different mode. So I want to show these slides that how we develop a project. Okay, you guys were developing a project, so it was good that I did not show it yesterday. So now you think about how you are going to develop a project. This is all my work, and Dr. Zain used the word serendipity. Okay. So this is all about serendipity, and we are very, very successful in developing some molecule which are really good. Let's go quickly. Introduction. Serendipity. What is the meaning of serendipity? It is happy accident or accidental discovery. You can call it. Refers to chance discovery, making discoveries by accident and sagacity of things which are not you are looking for. You just see that it happens. Okay. It is like that. And this was coined by Horace Walpole, 1754. The modern day therapeutic discovery is serendipitous drug, including empirical discoveries. It is not always unsought. So serendipity is a built-in drug discovery. And if you see the next slide, how many drugs are serendipitous? You can see that once you come up. So I can explain. This is a little bit older slide. It is about the same. Now, 2025 also, it is about 1450 drugs. Because I took this from this paper, okay? So that's why I'm putting there, but I know exactly. So about 437 drug molecules are on market, and approximately 24 have some element of serendipity, chance discovery, Somebody was making something, mixing something, and he got something, okay? So it became a drug. So this is called. The pie chart shows that how many there are, 75% is not serendipitous, and then others are. In case of cancer, it is more. That's what, because we talked a lot about cancer, okay? So in cancer, it is more, so there are 64% are non-serendipitous, and there are 14% are serendipitous, and derivative of serendipitous are there. So 88 drug molecule all market, 13 are serendipitous, 18 are chemical derivative of those 13. So there are about 35 clinically used cancer drug. So, we call it drug by chance or drug by luck. And we chemists always say, as a medicinal chemist, that we have to be lucky also. So for people like you are looking for Nigella, right? Sativa, you have to be lucky. So you need to pray a little bit, okay? So we do that, okay? So it happens. Okay, so example of serendipity, I think one of them was given, but I thought that I'll just give you a couple of examples. Penicillin was discovered in 1928, discovered by Sir Alexander Fleming, and it used to kill the bacteria, antibiotic or bacteria killer. And when he saw the mold on the plate, he said, it looks funny. That was his word, it looks funny, okay? Then there is a sildenafil citrate. This is a lifestyle drug. And then discovery of Lytic, which is used for the eyelashes, okay? Increase the eyelashes. And that was also discovered by chance. There is a book about that. Again, you can read that. And I met, fortunately, this person. And he has given a talk in our institute, Martin. So you can look at how, there's a whole list of serendipitous book there, a drug there. So you can look at that. So this is also part of our game, that we look for serendipitous. Now you will say that, oh, I'm talking about an old timer. No, I will give you a very new example. This is JAK inhibitor, COVID-19 inhibitor, baricitinib. This was discovered by chance also, okay? And in my family also, my sister had COVID, my sister-in-law had COVID, my father-in-law had COVID. Two of them at that time we get somehow from the Baroda, but my father-in-law could not get, so he passed away. So this is a drug which was meant for arthritis, okay? But it was used So it is written all there. Early in the pandemic, there's a company called Benevolent. So AI we talked about a lot. So they made this chart and they found out that this may work. Again, I am using word may work in that. So this is the pathway, and it is an anti-inflammatory drug, so it worked at that time. Okay. So this is the new serendipitous drug. Okay, what I have done, because I'm quite famous in our institute that I really I'm a believer in serendipity. So I thought that 4 or 5 slides I quickly do that. So I have a collaborator. His name is Professor Ravi Al-Kaisi. And when I came 10 years ago, and he was making photochemically responsive crystals, he makes some crystals which are energized, and he used to publish a lot of things. And I told him that, why you are doing that? Let's work on some anti-cancer compound or some anti-tumor compound or anti-ulcer compound. He said, no, I don't want to get into that. My area is focused. I just want to do that. I have done my PhD, postdoc, 2 postdoc. I don't want to go there. So somehow I convinced him and got some compound from him and got it tested, because we have Dr. Aziz was also there. So we tested MTT, we call it MTT assay. It became a full project. I was funded about 1.2 million real for that. And I will show you how we got there. 5 more slides. Because it was long presentation, so I cut it short so that you can understand. So I told Dr. Kaisi that you are making some compound, which you say that it will not be a drug, but look at this paper. When I showed him, this is the compound on your right is explosive. It is used in satellites and other sending satellite in the. So I said this compound was done by this compound was synthesized by some people, and they are doing clinical trial. It is in phase 3 clinical trial. And it was from energetic compound to very nice anti-cancer compound. And when I showed him paper, he was still not convinced. He said, no, I'm not doing anything drug discovery. No, I'm not going to do any drug discovery. So it was tough for me to convince him to get some drug from him. People are very. So when I showed this paper, then he was a little bit soft. I said, okay, I'll give you some compound. So that's what I wrote, that we developed a new class of anti-neoplastic agent from photocally responsive crystal to anti-cancer compound.
Academicians love to see
Publication, yes. And from me, he got first patent, which I'm going to show you, and he is very happy.
That's a different level.
Yeah. So anthracene-based photochemical responsive compound, so this was the preliminary result. I will not read everything. Just I want to, these compounds cannot be a drug, okay? These compounds, they are very greasy compound. They will not cross blood-brain barrier. They will not cross, cell membranes. So when I saw that, I said, give me, Professor Kaisi, couple of compounds. Let me test. So we tested some compound. I'm not showing that slide. And it was active. A good amount of activity we got on. Then we keep on, optimizing. You hear the word optimization a lot. So we keep on optimizing this molecule, and we got 2 surprises, which you'll see on next slide, and they all are patented. So what we did, now from below there was a 3 ring. Okay, those who are not chemists, that was 3 rings. So we changed, I designed molecules, we changed the rings, we changed the upper part also. So if you see the compound on your left, first compound, that was in MDA 237, this is a triple-based cancer cell line, and HCT is, remember, HCT is a colon cancer cell line. So it became 1.37 micromolar. We changed the nitrogen, so this is optimization. That's what I want to cap up, the optimization part, that how chemists think. When we switch the nitrogen from there to there and switch the connection, you can see that MDA 237 is 1.375 and HCT is 12.3. So we got an element of selectivity within cancer. Number one, we got a compound which is equivalent to mitoxantrone, which is already marketed drug. So my objective was complete. I can close the project now because that was the objective. But no, we went further. We got this compound, and this was, we call it dances with nitrogen. So this was like between me and him. He was very excited, and then he said, I will change the upper part, and he did many things, and we got a very good compound. By yeah. So we did not change any nitrogen because we got enough nitrogen there. Solubility was not a problem, but nobody used the upper part. That was in literature, we could not find anything. So we got it patented also. And this was, I think, the first patent. Yeah, one more story. So when we are doing this work, we saw a paper coming from from the UPenn, University of Pennsylvania, very close to our work. I said, who stole our work, right? So look at this molecule, and I will again click one more time, then you will understand better. So I just made these molecules. Look at that. These molecules, phenyl, thiophenyl, naphthylfuranil, but it does not have the anthracene. So that's okay. We are not going to go for anthracene. We made something else. We changed the upper part to the what I showed in previous, which is called 1,4-dioxylane. So we immediately went into action, and we immediately went into action, and we patented our molecule, and this became first-in-class dioxylane compound. We got the US patent. We got the Saudi patent also. We were very fast. We confirmed, and this later we did some mechanistic study, and we found it is a topoisomerase inhibitor, which is a target. I think people were talking about target yesterday, so we did some work. It is very specific for breast and colon cancer. Easy scale up. We patented, so we have a robust protection. And we talked to some company, and hopefully they will be licensed. But we don't have the animal data, so we are going to get the animal data soon. Okay. Everyone was talking about the technology and AI and this and that. So this is, I'm just trying to convince you that technology is good to use, but sometime you have to think about this graph, okay? Technology goes right up, and we are very happy. Then suddenly it does not work, then it comes down, then it becomes plateau, like that. That is where we should work. I will leave this slide with you. You can read all those things, and there is a reference there also, okay? So technology, it is good to use all the time, but it is not always that, oh, AI is going to solve the problem. Dr. Zain was very nicely, he was telling all the time that I want to see a drug by AI. You have to take this graph with you, okay? This is what it says, hype. Go on the people. What we call it, dhadam, right? And then it will plateau. I'm not saying don't use AI. I'm not saying don't use, but be very, very careful, okay? You have to put your all effort into that, but think about outside the box also. So I'll leave this hype cycle with you. There are minute things are written there. I think it is worth reading for everybody, okay? So this is the, I think, the last slide, I think. And my friend, because I always tell about Professor Casey, how he became a believer in drug discovery. Before he was not a believer in drug discovery. And now we meet every day, 2:30, we bring coffee together, and he is the one who writes on anywhere, anything, on paper, on glass, in bathroom, on tissue paper, everywhere he writes. And he is working with me, and he's doing his work, and he's discovering more drug. And he has a very, very nice paper and patent who is going to publish in JACS. So JACS, yeah. We already published one with him, and we are going to working on the publishing another paper in JACS. So he knows more medicinal chemistry than me. He knows more kinetics than me. He knows more enzymology than me, because in 10 years, he read a lot. So take home message for you, read a lot, okay? This guy was not ready to do anything other than what he is doing. Very stubborn. He's an Arab, so it is very difficult to convince him. But I convinced him to do this, and now he is with me, and he is moving to actually University of California, Riverside soon. And this is me and him. This is an iconic picture. We are standing in a conference, and he was having coffee cup, and then he said, "Oh, can you tell me about this?" And this photo was circulated all over, in Saudi Arabia, looks like. So we call it iconic paper, that he can write on anything, chemistry. So that's what. So I thought that this is my final slide, and I wish you all the luck. Thank you for getting me here, to organizers, to Dr. Azhar, and meeting all of you. Whatever I know, I could impart, give you, and wish you all the luck for your future endeavor, and please be in touch. Thank you so much. Any questions? I'm still here. I'm not in the car. Okay. Anything you want to know more?
They want you to stay.
I think that will be possible later, not today.
This is the time you can shoot your questions.
Yeah, this is the time you can
I will
shoot.
be around, but these two people, they will not be.
Uh-huh.
So please utilize.
I will be just an email away from you, WhatsApp away from you, okay? And I will really like to help and see some of the ideas which you discussed yesterday, how you connect with industry. I will say one more thing. You made a group. This group of the drug discovery scientists now. You are a drug discovery scientist now for me, okay? Because the way you presented yesterday was incredible, out of my mind, okay? So you make a group, you make and work on 1 or 2 projects together, okay? Just write up something, send it to Dr. Azhar. Maybe he will send it to Dr. Sayyad or to me, and we can connect you with some people who are really good in assessing whether it will be viable or not. So this will be my idea to them.
I think that's an excellent idea.
Yeah.
Because we had 48 participants, right? And we can see that some of them, they were really, I can say some of them were not sure why they joined, but most of you who were interested in and you went along the whole course, we can work together at least to write something, right? Kind of a review or a meta-analysis or something. And Dr. Imad, Dr. Zan, we can be all together.
Yeah.
So shoot me your interests, that then we can work out a structure and go from there. Because I know in academia, whatever you say, even I have one slide, that whatever you say, publication is something we always crave for. There's a craving for publication. Everybody wants to publish, right? And it does help in academia.
No, we published also, but we patented also. I showed you both the thing, okay? Yeah.
So I think that is something that you can see coming out from this course, that you have got a tangible outcome, that you have a paper or publication. It doesn't matter, I mean, it could be several authors. Authors is not a limit, because now even I hope that more people would be contributing more, and it can increase the quality of the work.
It will be really nice idea if you sit down together, or few of you sit down together, think about a target, read the literature, write a review, and
Yes,
that will be,
I think,
yeah. I know you have not done any experimental work, but you can write a review without experimental work, and you can say that we learned from this course, so we are going to do that. That will be
Yeah, I
really
think
good. This
that's
will
an
be the
excellent
best
idea.
outcome of your effort here.
And who knows, that may be a primer for some future projects,
Yeah.
right? So while working on it, you might find some smaller subgroups who can work together and have some projects for your masters or your PhDs or postdocs or whatever.
I remember there were topics about neuroscience. There were topic about CDK1 kinase. There were topic about Nigella sativa, and then there were other topics. So, if you think, and it is not necessary that you stick to that. If you think something else, talk to experts here. Talk to us, and you can at least put a one-page summary that you want to do that. That is not a big job. Then we can work on it.
Yes, we can work on it.
We
That's
can
an excellent
define
idea.
that.
I think
Mhm.
I'll take it forward,
Yeah, inshallah.
and we
Yeah.
can include in our recommendations as well.
Okay.
So what other things that I would say, you would say in academia while they are here, and if they want to move to an industry, what would be the do's and don'ts that they should be looking
So in industry, you will be bound by the university rules, okay? But never, you can ask that, but you never give up. You can put your idea to the management or somebody. They want you to work for their specific job. That's why they are paying you. But you keep putting them, and you ask that, I'm working 90% time I will work on your work, but 10% time I want to have some idea and I want to discuss. I'm not doing anything, but this is what I want to do later. So tell to your supervisor. You have to be persistent, because initially you will hear no, and again you will hear no. But you go for 3-4 times, you will hear yes. Let's see what is there. Okay, there you should start doing that. So that's the.
Sir, now are you having some nanoparticle based drugs?
Mhm.
Sir, any drug have you worked, sir, where you are using nanoparticles for that? So I don't know, sir, is the nanoparticle being used for the drug discovery?
Mhm. Yeah, it is. There are nanoparticles already in use as a medication. Doxorubicin nanoparticle are being used. That's your question, that it will be used or not.
Has it been used already?
Yes, they are not one of them. There are many of them. Just Google it and you will find many nanoparticles are already there. Which one is there? Lipid nanoparticle is there, iron nanoparticle is there. That you have to figure out what you are going to do.
Actually, when I was in an MTech nanotechnology, so our sir, he said that doxorubicin, when it is capped with certain anisotropic nanoparticles, which are not circulating and safe, so they have absorption spectra in both, and ani region also, and absorption peak in two region. So by that, what we can do, we can target drugs to the tumor region, and we can add radiation. With that, we can heat up that cells. So we can attack that cell from two ways. So is that anything that you have come out?
Everything is possible. I think you have to design in a way that if you are giving as an injection, you cannot inject anything in the tumor. It is not going to work that way, because tumor may be already spread out also. So you have to design nanoparticle in a way that first thing, like solid tumor especially, they are hypoxic. So you have to design something which reaches there, bind into that, and in hypoxic or oxygen-less atmosphere, it is cleaved. So that's
If I understand your question correctly, he's asking if there's any nanoparticle which is serving as a drug. Because usually
No.
nanoparticles have been
It's
utilized
a
as
carrier.
carriers.
It is a carrier,
As
not as a
packaging
drug.
material.
Yeah.
I
That
don't know
one.
about any nanoparticle that is used as
That
a
one.
drug.
Actually,
Was that your
nanoparticle.
question?
Yes.
Yeah, not nanoparticle alone it is not a drug. It is a carrier. Actually, nanoparticle alone is a question. It may be toxic also.
Yes.
So I thought that you were asking that nanoparticle with drug. Nanoparticle is always used with drug or as a carrier of the drug.
That was also one aspect, sir. I'm saying, sir, with the drug, we are adding a receptor, we are adding a molecule by which we will target that tumor, and that we are adding that nanoparticle. For example, with doxorubicin, we are adding a lectin or receptor, or which we will target in the tumors, some receptors or some targets, and with that we are attaching the.
So you are just adding that. So it
nanoparticle.
is excipient, what you call it. Okay. Excipients are already built in the molecule or the cancer molecule. So I'm still trying to understand, because see, nanoparticle contains a drug. It is encapsulated, it is on the surface, inside the surface, outside the surface. There are many things are there. But doxorubicin, like example, there is a nanoparticle based doxorubicin. which has a better half-life, and it has a low toxicity. Why? Because it is encapsulated.
Modulating the drug.
Yeah, yeah. So you have to always.
Sir, is it possible as a bachelor student to write a project or enroll in a project?
Which period?
In a bachelor, pursuing a bachelor, is it possible to us to join any project?
Doctor,
I can take the question. See, in your bachelor's, it's mostly classes and projects, right? You can be involved, but it will be shorter. I don't think you have a dedicated last semester that you can do a project. So you can utilize that part of your curriculum that allows you to do a dedicated project for 6 months.
There is no curriculum in our course. There is no research project
Yeah, so
in our course.
to do your graduation first, so you have to follow the curriculum.
yeah.
Not to do a project and fail in your
Don't fail there.
in your
Sir, but if we have idea, some novel idea, and discuss with other, is it possible to join as a officially in a project?
As I said, if your curriculum allows that, you can't miss the classes, you can't just do research and while your classes are going on, your attendance is short. Let us talk about it later on. It's
Yeah.
an internal issue. How to take that out.
Anything else? Okay, thank you so much. And I have to. Anybody else? Okay. Okay. Good luck. My best wishes, and I will